So you got to look at the big picture, and we don't really spend too much time on the little pictures, although you just need to know what goes into each process and what comes out of each process, whether it uses oxygen or not, and where it takes place in the cell, whether it's cytoplasm or the liver, which we're going to talk about, or it's going to take place in the mitochondria. Okay? So again, the big picture. You learned from high school that if you took biology, you just knew the mitochondria is the powerhouse of the cell. It makes ATP. Well, now in college, we need to see, how do you make ATP? How does the mitochondria do that? That's the difference between high school and here. Okay? So we have two things that we need to make ATP. We need glucose, some sort of carbon source, okay? And you need oxygen. Now, we can make ATP without oxygen, but as you've seen, it's not effective, not too much, okay? So we have different forms of organic substances. That's what we were talking about, where carbon was coming in, that we could actually make energy with. The three forms of carbon structures that you eat is going to be carbohydrates, i.e. sugars, and that includes starches, right, long chains of sugars. We have fats, the triglycerides and all those lipids and cholesterol and stuff. And then we have proteins that are made from amino acids. We can actually make energy with these three items. So you got to always be eating, not eating like me, but I mean eating because you got to get that energy. So what I showed you last week was just one form of that. In other words, one source, the sugars, the so-called carbohydrates make an ATP. And we use glucose as our source. Okay? We're going to talk about when the fats come in, how do you make energy with that, and also a little bit about proteins. Not too heavy on those things, but we did talk about glucose. So this is just a quick review for you guys. This here is the cytoplasm of a cell. Over here is the mitochondria. The mitochondria can't be used unless we have oxygen. You need a backstage pass to get into the mitochondria. That backstage pass is oxygen. Okay? So we have a process where we're going to take glucose, which is made up of 6 carbons, and it goes through 10 different steps to make something called pyruvic acid, or also known as pyruvate. What this does is it breaks that into two things. We have 2 3-carbon structures. There's one there, and there's one there. Because this is just too big to get into the mitochondria. So we make 2 3-carbon structures. They're called pyruvic acid. We call this process glycolysis. Break up the word. Glyco, glucose, lysis. What does lysis mean? Break up. See? The names kind of tell you what's going on. In this process of going from glucose, a 6-carbon chain, to 2 3-carbon chains, 2 ATPs go into it, but 4 come out. You have to invest 2 ATPs to do this. It's kind of like you want to make a pottery shop, right? Someone has to give you some money. You go to an investor, they're going to give you, I'm just going to give you numbers here, they're going to give you $2 for you to go buy clay. You're going to buy the clay with $2. You're going to make figurines with it, maybe 2 different figurines. And now you could sell each figurine for $2 each. So you made $4. Does that make sense? But someone invested $2 in it. So really, net-wise, you only made $2. Does that make sense? So we have to put 2 ATPs into this 10-step process, but you get 4 ATPs out of it, which means you only make net 2 ATPs without oxygen. Okay? You also make something called NAD+. NAD+ is an electron carrier molecule, or it can accept electrons. It will accept one electron here to become an NADH. The H is the electron, the electron there. Because an electron has a negative charge, we're going to go from positive to neutral. The charge gets reduced, does it not? That's why we say the NADH is the reduced form. This is what we refer to as the oxidized form. Okay? Now this NADH is going to be used later when we have oxygen to make ATP. We'll get back to that. Now, the pyruvic acid, if it has oxygen, it will be allowed to come into mitochondria. If it doesn't, we're going to be talking about a process over here happening in the cytoplasm. But let's say it does have oxygen. One of these structures, we'll say that just one, and just follow it that way. One of these pyruvic acids would enter here. Both of them do, but it's easier to follow when there's only one. And then you'll multiply everything by 2 if I ask you what one molecule of glucose does. So one goes into here with oxygen. One of the carbons goes off here and combines with 2 oxygens. There's one carbon that comes off, leaving us how many carbons on one? 2, right? So this acetyl-CoA is a 2-carbon structure. Following? This 2-carbon structure is called acetyl-CoA, and this can enter something called the citric acid cycle, or also known as a TCA, the citric acid cycle, or Krebs cycle, same thing. All that's the same thing. And that's where it's going to meet up with this oxaloacetate. Oxaloacetate is a 4-carbon structure. The 2-carbon structure and the 4-carbon structure combine in a different way. It doesn't look like, it doesn't change into glucose. It turns into something called citric acid. Same stuff that you drink with orange juice. Bless you. And there's this cycle that goes around here that there's 8 different reactions, which again you don't have to worry about. But what you do have to worry about is what goes into the cycle and what comes out of the cycle. For every 1 molecule of acetyl-CoA, you get 2 carbon dioxides that come off of this. You get 3 NADHs. You get 1 GTP. For your purpose, just understand it's just ATP. And you also get 1 FADH2. This is where the money's being made. For every NADHs, you make 3 ATPs. For every FADH2, you make 2 ATPs. This is where you're making a lot of money. Bringing your figurines to the marketplace. You'll be making a lot of money there at the mall. Okay? You probably even up the price a little bit, right? These are carrying electrons. The H there. The H2 over here. They're carrying electrons. These electrons are going to be used in the electron transport chain. All the NADHs from all the places that they're being made are going to enter the electron transport chain, as well as also the FADH2s. and it's going to transport the electrons. And it's a set of reactions. It's not just one reaction. It's a set of redox reactions. Things get oxidized, reduced, and so forth. So it has all these steps. And as it does this, the last part of the chain is that this electron is going to be attached to oxygen. The oxygen is going to attach to two hydrogens, and you make water. We call this metabolic water. This is where water comes off of this whole thing, and carbon dioxide. Those are all the exhaust that kind of comes out of your engine. We don't really use, well, we use a little bit of the water, but carbon dioxide we really don't use. It just comes out of our breath, and trees use it, which we'll talk about photosynthesis. For every FADH2 enters over here, you get 2 ATPs. For every NADH that enters in here, you get 3 ATPs. Okay? That's a fast review of this. That's when we use glucose. All right? Now just keep in mind that I only talked about one acetylcholine that comes out of here. But if I ask, hint, on an exam, if I ask one molecule of glucose with oxygen is going to enter here, how many molecules of carbon dioxide are made total? Think about it. Don't answer right away. Total. One molecule of glucose makes how many molecules of carbon dioxide if we have oxygen? There's 2 here. There's 1 here. How many is that? 3, right? But that's 1 pyruvic acid that comes in. We're going to have 2 that come in here. So then how many carbon dioxide are made here? 2. How many are made over here? 4. So how many molecules of carbon dioxide are made from 1 molecule of glucose if we have oxygen? 6. Do you see how to do the math? Don't memorize. The only thing you got to memorize is this thing. But understand the concept. My suggestion when you take this exam on Thursday is take one of the blank sheets that are someplace in there and draw this whole friggin' thing out. Make your life easier. Instead of you trying to guess all this work, you can now have a picture on there that you could actually relate to. It's what I did in college, it's what I did in medical school. Does it take 10 or 15 minutes away from the time that you need to take the exam? Yeah. But you don't have to think about it. You just have to flip to that page and look at it. So my suggestion is from now until the day that you take the exam, draw these pictures out 2 or 3 times a day. The basic formula, I wrote it to a few people here. Actually, I told everyone about this, but I want to emphasize it to you guys if you haven't read my email. The basic formula for biology and A&P, anatomy and physiology, is for every 1 hour you're in class or lab, that's 3 hours you're at home. And you got to do effective study. What that means is, well, in the summer we're spending 14 hours here a week, which means you should be putting in 40 hours at home. If you're only putting 16 hours, it might match the reason why you're getting certain grades on there. If we do this in the fall or spring, you only need to spend 14 hours a week. It's just the rule of thumb formula. Okay? All right. So do you understand this? If I said, if we have 3 bouts of acetylcholine, acetyl-CoA entering the tricyclic, or the citric acid cycle, if 3 bouts of acetyl-CoA enter that, how many NADHs are made? 9. Does that make sense? All right? It's not hard. It's easy once you got this picture up there.